Good morning. Thank you for joining us in today's webinar. I'm Nancy Kiao, a board-certified pathologist trained in anatomic and clinical pathology, and I work in the Pharma Assay Services Group at Advanced Cell Diagnostics. Our group supports investigators with assay development and scoring of samples that have been stained with RNAscope. In today's webinar, I will cover RNAscope data interpretation. Before I begin, I would like to mention that you can enter your questions into the chat box in the right-hand side corner of the WebEx screen. Unfortunately, you will not be able to ask the questions live on air during the session. However, we will answer as many questions as we can at the end of the presentation, and the ones we missed will be answered offline. Thank you again for joining us. Here's the agenda for today's webinar. I'll briefly go over the RNAscope technology and give a few examples of RNAscope applications before getting to the heart of the webinar and discussing RNAscope staining and scoring, which will include comparisons with IHC, looking at signals, reviewing definitions, then touch on the sensitivity and limit of detection of RNAscope. Then I'll go over manual scoring systems and finish this section with image analysis and quantitative scoring. Finally, we'll look at QCing RNAscope results. At the end, there will be a Q&A session. We begin by going over RNAscope technology. The detection of protein with immunohistochemistry, or IHC, is well established and routinely used in clinical practice and research, as is FISH for DNA detection. And while quantitative RT-PCR is commonly used for RNA detection and quantification, before the commercial launch of RNAscope in 2010, there was no robust and sensitive method for detection of RNA within intact cells. With that, we will review RNAscope technology. The RNAscope assay begins with a unique double D probe design, which we will look at a bit more in depth after this overview slide. RNAscope works with a variety of cells and tissues, the most common and clinically important being formalin-fixed paraffin-embedded or FFPE tissue. As with IHC, the tissue is placed onto a glass slide where the cells are permeabilized and target mRNA is unmasked. This is followed by hybridization of a pool of about 20 target-specific double Z probes to target RNA and amplification of hybridization signals via sequential hybridization of amplifiers and label probes, resulting in detection of single RNA molecules in single cells as punctate dots in their morphological context. These dots can be viewed under a standard microscope and quantified on a cell-by-cell basis manually or with image analysis software. As I mentioned, the RNAscope assay begins with a unique probe design for the detection of target RNA. The probes are Z-shaped oligonucleotides composed of an 18 to 25 base sequence lower region that's connected to a 14 base tail sequence upper region via a spacer. When a pair of Z probes hybridize next to each other at a target binding site, which spans approximately 50 bases, the upper regions of the two Z probes, now next to each other, form a pre-amplifier binding site of about 28 bases. This probe design strategy is similar to fluorescence resonance energy transfer, or FRET, as two independent Z probes have to hybridize to the target sequence in tandem in order for signal amplification to occur. For each target RNA, a pool of about 20 double Z target probe pairs spanning about 1,000 bases are designed to specifically hybridize to the target molecule, but not to non-targeted molecules. Once the pre-amplifier binding site is formed, signal amplification can take place. Similar to branched DNA assays. An L-shaped pre-amplifier molecule is added, which only binds if it sees a pre-amplifier binding site. Amplifier molecules added in the next step bind to multiple binding sites on the pre-amplifier molecules. Finally, labeled probes containing fluorescent molecule or chromogenic enzyme bind to the numerous binding sites on each amplifier. The result is a Christmas tree of sorts with about 400-fold amplification per double D probes. Since we have a pool of 20 D pairs each with 20 amplifier molecules which each bind 20 labels, this means that for a 1 kilobase target region, the amplification strategy can recruit up to about 8,000 labeled molecules. However, if a Z probe binds to a non-specific target, it is unlikely for the adjacent Z probe to bind as well to form a pre-amplifier binding site. Therefore, the pre-amplifier will not bind to a single Z probe, and no signal will be generated. Thus, the double Z probe design provides a highly sensitive and specific method for RNA transcript detection through signal amplification and suppression of background noise. It has been shown that binding of only 3 of 20 double Z probes to target RNA is necessary for the detection of a single RNA molecule, such that employment of a 20 ZZ probe pool provides robustness against partial target RNA accessibility or degradation, which would be encountered with formalin-fixed paraffin-embedded tissue and with targets with bound proteins as seen in this diagram. The RNAscope probe design strategy is thus approximately 100 times more sensitive relative to traditional RNA ISH methods, which utilize single deoxygenin labeled probes, which you can see on the right-hand side of the slide, compared to RNAscope on the left for the detection of SIV in lymph node tissue. Briefly, RNAscope can be performed on, as I mentioned earlier, FFPE tissue as well as fixed frozen tissue, fresh frozen tissue, and cell pellets, either on manual platform with the red-brown chromogenic singleplex, brown-red or green-red chromogenic duplex, or multiplexed fluorescent assays, or with automated platform, also with singleplex, duplex, or multiplex fluorescent assays. RNAscope uses universal assay conditions. Every target-specific probe uses identical hybridization conditions. Specific kits with common reagents and optimized protocols have been designed by APD for use on the LICA Bond RX and Ventana Discovery research platforms. However, the RNAscope singleplex assays with the brown chromogen are available for purchase through LICA and can be utilized on the LICA Bond 3 clinical platform, which is identical to the LICA Bond RX in terms of hardware, but differs in software packaging. The automated RNAscope platforms will give similar results as compared to the manual assays, but save you time and allow for greater reproducibility. Let's take a look at some RNAscope applications before going into RNAscope scoring. The use of RNAscope technology has grown almost exponentially. As of this month, it has appeared in more than 800 peer-reviewed publications such as Nature, NEJM, the American Journal of Surgical Pathology since 2011, and in a wide variety of fields from oncology to infectious disease to developmental biology. As we saw on the last slide, RNAscope most often appears in oncology publications, especially as PD-L1 and other markers for companion diagnostics is a hot topic these days. This is an image of non-small cell lung carcinoma that has been stained with an RNAscope green and red duplex chromogenic assay. The lung cancer cells express PD-L1 as evidenced by the green signal in the tumor cells with larger nuclei. Scattered throughout the tumor are CD8 positive cytotoxic T cells marked by the red signal, most of which do not co-express PD-L1. However, there are occasional cytotoxic T cells with PD-L1 co-expression, indicated here by the orange arrow. RNAscope is a powerful tool for detecting and localizing RNA in cases where no antibody does or will exist, as is the case with long non-coding RNA. These are non-coding RNAs that are longer than 200 bases, some of which have been shown to regulate gene expression and have roles in tumorigenesis and other diseases. In this study from the University of Michigan, the authors used RNAscope to study the expression of a long non-coding RNA called SHLAP1 in prostate cancer. They found that SHLAP1 expression increases with prostate cancer progression from low Gleason score to high Gleason score to metastatic castration-resistant prostate cancer, and localize the transcript to the nucleus. Whereas the last two examples employed a chromogenic RNAscope assay, this image is an example of a multiplex fluorescent assay, but using only 2 channels rather than our current maximum capability of 4. Alternatively, 3 RNA markers and 1 IHC marker can be interrogated. This sample is peripheral blood from a breast cancer patient stained with pan-cytokeratin and CD45. The CD45 RNA molecules in the white blood cells appear as red punctate dots, while the cells with green signal are the circulating tumor cells with high expression of pan-cytokeratin, and RNA molecules seen as clusters and clumps. The detection of high-risk human papillomavirus, or high-risk HPV, is clinically important not only to determine an etiology of a malignancy, as is the case with oropharyngeal squamous cell carcinoma, or high-risk HPV positivity would mean a better prognosis due to less aggressive behavior, but also in making a diagnosis of neoplasia in diagnostically challenging anogenital lesions. In a very recent study by Mills and others that appeared in the American Journal of Surgical Pathology, they performed and validated, notably on an automated platform, the LicaBond RX, an RNA ISH assay using RNAscope technology and RNAscope's HPV HR18 probe, which detects E6, E7 mRNA in 18 high-risk HPV subtypes against PCR, DNA ISH, which is now no longer commercially available, and P16 IHC in 102 samples of cervical, vulvar, anal, and head and neck neoplasia. The results again show that RNA ISH with RNAscope has high sensitivity and specificity for HR-HPV in head and neck cancers, but now on an automated rather than manual platform. The results also showed high sensitivity, not only in high-risk HPV-related oropharyngeal squamous cell carcinoma, but also greater than or equal to 97% detection for the HR-HPV in anogenital high-cell and cervical squamous cell carcinoma as well. These are only some of the many applications of RNAscope technology, which will provide highly sensitive and specific molecular detection in morphological contexts. We turn now to RNAscope staining. This chart compares IHC with RNAscope, which share many similarities in terms of modality, tissue type compatibility, and performance on cell type. Both utilize chromogenic or fluorescent assays. Both can be performed on FFPE tissue, frozen tissue, and other tissue types. Both can be performed at any cell type. In contrast to the generally diffuse staining with IHC, RNAscope signal is typically seen as punctate dots and clusters. Also in contrast to IHC, where the subcellular localization of signal varies with the target, With RNAscope, the signal is usually in the cytoplasm, where transcript is transported to, with occasional detection in the nucleus. Some of the common IHC staining patterns are shown in these images and listed here, including nuclear, cytoplasmic, membranous, nuclear and cytoplasmic, and membrane and cytoplasmic staining. Other staining patterns are also possible, but less commonly seen, including specific staining of subcellular compartments, organelles, and other specific areas of the cell. These two images show the contrast and staining pattern of RNAscope versus IHC with P16. On the right, with IHC, there is diffuse nuclear and cytoplasmic staining, while on the left, staining of RNAscope, the signal appears as dots and clusters in the nucleus and cytoplasm. Some questions you may have at this point include, what does a dot mean? What is the significance of dot size? What is the difference between a dot and a cluster? And what is the sensitivity of RNAscope versus the IHC? In this image, in which the RNAscope chromogenic assay has been performed with a red chromogen, signal appears as numerous punctate red dots of various sizes and intensities and few clusters. It is the number of dots and/or clusters that are important, rather than the size of the dot or the intensity of the dot. Each punctate dot equals an mRNA molecule. The small faint dot pointed by the orange arrow is one RNA molecule. The slightly larger, darker red dot, which is also very nicely round, pointed by the teal arrow, is also one mRNA molecule. However, its larger size is due to more double-D probes bound to the target molecule. In contrast, a cluster pointed by the brown arrow is more oblong with a slightly irregular border and results from overlapping signals from multiple mRNA molecules. Similar patterns of dots and clusters are seen in fluorescent versions of the assay. The orange arrows point to single mRNA molecules with less double-D probe pairs bound to them, compared to the dot pointed by the teal arrow. And the brown arrow points to a cluster with an irregular shape, rather than having a round shape, which has resulted from overlapping signals from multiple mRNA molecules. To reiterate, in general, RNA expression for a given biomarker is related to dot number rather than dot size or intensity. In general, dot intensity is related to the number of double-D probes bound to a target molecule. Therefore, cells with similar numbers of dots, but with different dot intensities or sizes, have similar levels of RNA expression. While RNAscope is a powerful tool, there are occasions when you may be faced with some artifactual staining that you may wonder about how to deal with it and/or what it means. As with IHC, there may be an edge artifact where there may be more signal at the edge tissue. Slightly seen here, the edges of this core tissue. Focus should be placed on scoring tissue slightly away from the edge if this edge artifact is seen. An endogenous biotin background, most common in the kidney and liver, results in tissue with a pink haze, almost reminiscent of eosin staining. But you can see that it is distinct from the true signal of punctate dots, here seen in red. While we don't yet have an official protocol to reduce this biotin background, ACD support can certainly help you with your concerns or questions regarding this. This is just to show you what red blood cells look like with RNAscope. Here is a blood vessel in lung tissue that is filled with golden stained RBCs. And finally, I wanted to mention that you may encounter red chromogen trapping in adipose tissue. I apologize for not having an image to show you, but this is not true signal, as the red chromogen will be very finely granular as opposed to punctate dots seen with true RNAscope signal. The RNAscope detection limit is typically below the detection limit for IHC. These are images from 3 different MET cell lines, the first 3 columns of which have been stained with RNAscope probes, DAP-B, a negative control marker, Polar 2A, a positive control marker, both of which we will discuss later, and CMET. The rightmost column is MET IHC. The low-expressing MET cells in the top row are negative for MET by IHC. However, CMET RNA is detected with RNAscope, and protein was detectable by Western blot. Could the RNAscope CMET signals be background noise rather than true signal? Well, re-examining the negative DAP-B control stain slide here in this field that has no staining whatsoever reassures you that the signal in the CMET stain slide is real. And RNAscope technology was able to pick up low-level expression that IHC was unable to. In fact, RNAscope is as sensitive as RT-PCR. In experiments looking at HER2 expression in formalin-fixed paraffin-embedded tissue, the number of dots per cell with RNAscope was highly correlated with transcript levels from RT-PCR. We turn now to manual RNAscope scoring, which includes qualitative assessments, semi-quantitative assessments, and H-scoring. While high RNA expression in cells can be seen at 10x, 4x, or even 2x, we recommend assessments be made about expression levels at least 20x magnification. Usually 40x magnification is necessary to count dots and assess tissues that are low expressors. Qualitative assessment with just a positive or negative for the presence of the marker is the simplest scoring method and could be appropriate for those that are highly expressed, as is the case with albumin in hepatocellular carcinoma, seen in the left panel with so many dots in the cytoplasm that it is comparable to moderate to marked cytoplasmic staining with IHC. On the right panel is the same tissue stained with DAP-B, a negative control marker that is completely negative. Also, qualitative assessment with just a positive or negative for the presence of the marker would be more important than quantifying expression of a marker in certain clinical situations, as would be the case with cytomegalovirus or CMV chorioamnionitis, seen in this image of chorionic villi with CMV infected cells. Seen in this table is the more common semi-quantitative scoring method employed for IHC, where negative staining is given a score of 0, while intensity or amount of staining is broken down into 3 categories of weak, moderate, or strong, with a corresponding score of 1+, 2+, or 3+. For example, this is what is routinely done in clinical practice when assessing nuclear staining of ER and TR in breast cancer cases with the addition of noting the percentage of cells with the given score. A similar quantitative scoring system, shown in the table here, was developed by ACD's R&D department and used in the Pharma Assay Services group here at ACD. This is a 0 to 4 system based on the number of dots per cell, again, not on dot size or intensity. No staining or less than 1 dot per 10 cells is a score of 0. 1 to 3 dots per cell is a score of 1. 4 to 9 dots per cell with no or very few dot clusters is a score of 2. 10 to 15 dots per cell and or less than 10% dots in clusters is a score of 3. Greater than 15 dots per cell and or greater than 10% dots in clusters is a score of 4. The images here give you an idea of what each score may look like. In the Pharma Assay Services group, we've generally been assigning a single score based on the predominant non-zero, obviously unless it is a score of 0, staining pattern seen in the sample, unless it's clear that different cell populations have different expression levels. The investigator has specific requests or evaluation by a pathologist is requested. We are also transitioning to providing the percentage cells with the score. In cases with significant heterogeneous expression of a marker, we can also provide the range of scores seen. This is an example semi-quantitative scoring system only. Other scoring systems are acceptable. For instance, you may want to condense some of the categories. So you have the 1, 2, 3 that's more often used with IHC, or you may want to add more categories. Here's a sample stained with the DAP-B negative control marker having no dots, thus with a score of 0. In this sample, most of the cells contain from 1 to 3 dots, thus getting a score of 1. In this sample, most of the cells contain from 4 to 9 dots, with no or few clusters, thus getting a score of 2. In this sample, most of the cells, notably in the tumor cell compartment, contain from 10 to 15 red dots, thus getting a score of 3. In this sample, most of the cells contain more than 15 red dots, more than 10% of which are in clusters, thus getting a score of 4. Another semi-quantitative approach for scoring is H-scoring, which has been used with IHC and can be used with RNAscope. The H-score, or histoscore, is a method assessing the extent and variability of staining of a tissue for a given biomarker in a more continuous fashion. With IHC, H-scoring takes into consideration the staining intensity in conjunction with the percentage of cells staining positively at that intensity. So with the more common 4-category method of 0, 1, 2, 3, the H-score is calculated by the formula of 3 multiplied by the percentage of strongly stained cells, which are those with a score of 3, plus 2 multiplied by the percentage of moderately stained cells, which are those with a score of 2, plus 1 multiplied by the percentage of weakly stained cells, which are those with a score of 1, plus also technically 0 multiplied by the percentage of negative staining cells, although this portion will always be 0. For a 0 through 3 scoring system, the scores can range from 0 to 300. With the 5 categories of 0, 1, 2, 3, 4, we routinely use with RNAscope, the maximum H score will increase by 100 to 400. The sample can then be considered positive or negative for a marker on the basis of a specific discriminatory threshold value. The next section is on image analysis and quantitative scoring. Whole slide scan images, or images taken from a microscope camera, can be analyzed by a number of commercial image analysis software packages or by free open source programs. Briefly, with respect to the free open source programs, the most widely known is ImageJ. The newer version is called ImageJ2, which is written in Java, so it can be used on Macintosh, Windows, and Linux platforms. Another is called Fiji, which stands for Fiji is just ImageJ. Fiji describes itself as a batteries included distribution of ImageJ, bundling a lot of plugins which facilitate scientific image analysis. Our Pharma Assay Services group at ACD has both SPOTSTUDIO and HALO for our use. However, we pretty much use HALO software exclusively at this point in time. And while others in the group have some experience with SPOTSTUDIO, I personally do not, having only worked with HALO. SPOTSTUDIO is a custom-designed algorithm developed by ACD in partnership with the Definians that enables quantitative analysis of gene expression with single molecule sensitivity. It is compatible with RNAscope chromogenic singleplex assays only. HALO was developed by Indica Labs and works not only with RNAscope singleplex assays, but also with more complex expression data generated with the RNAscope duplex and multiplex assays. HALO provides interactive results with real-time readout, and the software also reports individual cell data and maintains an interactive link between cell metrics and cell imagery. This is an example of SPOTSTUDIO image analysis for HER2 and breast carcinoma via RNAscope. A region of interest has been manually drawn, demarcated by this thick red line. The software picks up cell nuclei based on hematoxylin staining and then identifies cell boundaries through approximation of average distances between the cell nuclei. Dots are detected based on pixel color. Single dots are outlined in yellow, while clusters are outlined in dark blue. The algorithm then counts dots within each cell and bins and outlines each individual cell by color based on the number of previously established categories or bins. In this situation, blue for cells with 0 to 1 dot, green for cells with 1 to 3 dots, red for cells with 3 to 6 dots, and orange for cells with greater than 6 dots. The results of the analysis for this region of interest shows that the majority of cells have at least 1 dot, with a little more than 30% of tumor cells containing 1 to 3 dots, and a little more than 30% of tumor cells containing 3 to 6 dots. Almost 25% of tumor cells have more than 6 dots, and about 10% have either 0 or 1 dot. In the panel to the right, you can see each cell is assigned a unique ID number with the number of dots it contains. In the panel above the histogram, you can see that some other information is generated for this region of interest, which includes total area analyzed and total cell count. As I mentioned earlier, not only can HALO analyze images from RNAscope singleplex assays, but also more complex expression data generated with the RNAscope duplex and multiplex assays. Again, the software picks up cell nuclei based on hematoxylin staining and then identifies cell boundaries through approximation of average distances between the cell nuclei. Dots are detected based on pixel colors. Halo allows for real-time tuning of analysis settings. The algorithm then counts dots within each cell and bins and highlights each individual cell nucleus based on dot count into the categories, or bins established. In the markup, darker shades indicate higher spot counts, while white nuclei indicate 0 dots. This is an example from an RNAscope duplex red-brown chromogenic assay with hematoxylin counterstain. The markup images show the Halo ISH module markup for dual and single probe detection. In the top left-hand field, most of the cells contain brown dots, and there looks to be a single cell in the upper left-hand corner containing red dots. Unlike the example I showed you for SPOTSTUDIO, I believe the binning in this example is in line with our standard scoring system. So looking at the dual probe markup in the top right, these cells with white nuclei have no dots. The cells with light green nuclei have 1 to 3 dots. The cells with slightly darker green nuclei have 4 to 9 dots. You can see that the software has identified the lone red cell with red dots. If there had been any cells of co-expression, the nuclei would have been highlighted with yellow or brown. A Halo can bin each and every cell in a region of interest based on the number of dots in that cell into a previously defined category. A single H-score can be calculated. This histogram is similar to the one that we saw earlier with SPOTSTUDIO. However, the y-axis here is number rather than percentage of cells. Over here is the percentage of cells in this field of view in each category and the H-score calculation. While SPOTSTUDIO also bins cells, the software does not automatically calculate an H-score. That must be done manually with SPOTSTUDIO. Halo not only provides you with the H-score for a region of interest, but it also generates a plethora of information, including but not limited to total area analyzed, total cell count, average dots per cell, and the percentage of cells with at least 1 dot. The final section today covers QCing of RNAscope results. Positive and negative control probes should always be run with your RNAscope assays. The 3 positive controls used with RNAscope assays are ubiquitin-C or UBC, DNA-directed RNA polymerase II subunit RPB1 or polar IIa, and cyclophilin B or PTIB. These are housekeeping genes expressed at similar levels in most tissues. Their use is analogous to that of beta-actin and GAPDH for RT-PCR in northern blots, and is for determining the RNA quality of the sample being tested. If signal is present above a certain threshold, the tissue is suitable for RNAscope. However, if it is below the threshold, the tissue is not suitable for RNAscope. ACD developed a universal negative controlled probes targeting the DAPB gene from a strain of Bacillus subtilis, a soil bacterium. The negative control is used to ensure that there is no background staining and that the tissue sample is appropriately prepared. If signal is present above a certain threshold, the tissue is not suitable for RNAscope. However, if it is below the threshold, the tissue is suitable for RNAscope. This RNAscope strategy goes beyond what is done with IHC, since with IHC, housekeeping proteins, at least in clinical practice, are not evaluated in parallel with the target on the same sample tissue to ensure that antibody detection is optimal. Depending on the target, different positive control probes are recommended. The most flexible, most commonly used positive control probe is PPID. In the vast majority of studies, if PPID is positive, then any target probe will detect the target RNA. For low expression targets, we recommend Polar 2A, and for high expression targets, we recommend UVC. The reason for this is that UVC RNA can potentially be detected with RNAscope, despite substantial degradation and under non-optimal assay conditions, leading to false negative results. Before running an RNAscope assay with target probes, your technique should be checked, and the sample should be qualified with the appropriate positive and negative control. In the first step, slides prepared from cell lines are tested with the appropriate positive and negative controls. If the PPID stain slide scores greater than or equal to 2, which is 4 to 9 dots per cell, and the DAP-B stain slide scores less than 1, the technical check passes and you can proceed to the sample RNA quality check, in which sample tissue is probed with the positive and negative control. Again, if the PPID stain slide scores greater than or equal to 2 and the DAP-B stain slide scores less than 1, the RNA quality of the sample is sufficient to proceed with testing of the target probe. If the QC check fails, you should verify technique, perform assay optimization by tweaking pre-treatment conditions, or check RNA quality with new samples. The following RNAscope example case will go through the steps of the QC process. In this technique check, UVC was used as the positive control probe on the cell line. As UVC has a moderate to high expression level, in order for a sample to pass, it must score at least a 3. So most cells must have at least 10 dots, which this cell line does. Also, the negative control has no staining, so the technique is good. Having confirmed the proper technique for running the RNAscope assay, we can now proceed to checking the tissue sample for RNA quality. The same control probes are used here, UVC and DAP-B. The sample passes with a score of 3 for UVC and a score of 0 for the DAP-B, indicating that the RNA quality in the tissue sample is suitable for RNAscope testing. Having confirmed that the RNA quality in this tissue sample is suitable for RNAscope, we can now test the tissue sample with the target probes. In this case, various HPV target probes. In these images, we are targeting several HPV subtypes, HPV 16, 18, and 31 to 33, which are high-risk HPV, and HPV 6 to 11, which are low-risk subtypes. As you can see, this tumor tissue is positive for HPV, seen with the brown dots, and negative for the other subtypes tested. An optional step 4 can be performed and is recommended if known positive and negative control cell lines or tissues are available, similar to IHC control strategy. In this case, the Cascade cell line, which is known to be positive for HPV 16, and the HeLa cell line, which is known to be negative for HPV 16, are employed to demonstrate specificity of HPV 16 probe staining. Here, the HeLa is completely negative, while the Cascade is positive for HPV 16. Once the technique is working reproducibly, labs can skip the technical check and run the tissue sample check and test probes at the same time. In this RNAscope singleplex experiment with brown chromogen, the positive PPIB control shows moderate levels of expression, while the negative DAP-B control is clean without signal. So the RNA quality is good, and you can be confident that the signal you see with your test probes is real. In this case, the sample shows high EBV expression with what looks like diffuse staining localized to the nucleus. I hope this webinar has provided you with some guidance that will help you with data interpretation of samples tested with RNAscope. This concludes the presentation, and we will proceed to the Q&A session. Just to note, if we do not get to your questions in time or cannot address your questions, please feel free to contact ACD support through email or phone. So a question that came in is, why did I not see signal with positive control? So there are several possibilities for this, one of which is that potentially the RNA quality was not so great and it was likely degraded. Additionally, sample preparation or pretreatment was not optimal, so you should go back and check your technique and tweak some of the pretreatment conditions. Another question that came in is, can I dilute my probes to reduce concentration and therefore my background signal? The short answer is no. And the reason why is the RNAscope assay kits that you have purchased contain reagents that should be used as they come. The probe concentration is optimal and no further dilution is required. So please contact technical support at ACD for guidance on reducing the signal. Another question is, I'm working with mouse kidney and liver FFPE samples. Some of the samples were fixed less than 12 hours. I understand that the signals in these underfixed samples may be faint. Are there modifications that I should make to the standard protocol? Your samples might require a reduced pre-treatment condition, likely typically target retrieval as well as protease. For additional support for your particular case, please contact technical support at ACD. So some more questions have come in. One is, I believe, the HALO related or image analysis software related, which is, how can you accurately outline cell boundaries manually? What is the criteria, or just use imagination? So the software, if it's easier to use, should be able to do that for you automatically. But if, as you're tuning the analysis parameters and you feel like these cells, you want them, the software to make the outlines larger or smaller at your discretion, you can certainly adjust the cell radius. There's in HALO, for instance, there's a parameter where you can do that, and that will change the cell boundary. Another question that came in also regarding digital imaging analysis. Is it possible to separate the dots from malignant cells from the dots in the inflammatory cells? Yes, it is possible. For instance, with HALO, and I hope I'm answering your question correctly. With HALO, there's a tissue classifier that we've started to use recently. And it's a really great tool, although it also does require some tweaking. But you can teach the software what a cell type is. So you can go in and sort of, with a few strokes of your mouse, say these are tumor cells, or these are stromal cells, or these are immune cells, etc. Whatever you feel like, anything you want to designate, you can. And then after you've taught the computer enough, the software enough, it will then, whatever field that you want to look at, it will classify the cells into whatever categories that you've established. So when you do your analysis, each region or each cell of interest you'll have separate H-scores for those particular categories. So thank you for joining our webinar today. As I mentioned, if we could not get to your question, we will certainly reach out to you offline. Thank you for joining us today's webinar.